Hello everyone, I am Ms.S.Veeralakshmi, Assistant Professor, Department of Microbiology, the Standard Fireworks, Rajasthan College for Women. Today we discuss about what is paper chromatography and the principle and methodology and their applications. What is paper chromatography? Paper chromatography is a technique in which the analysis of unknown substance is carried out mainly by the flow of solvent on specifically designed the filter paper. The principle behind the chromatography: first, the paper chromatography involves the partition chromatography or adsorption chromatography. Partition chromatography is the substance or partition or distributed between the liquid phases. In the adsorption chromatography, the solid and liquid phases involved, where the solid surface of the paper is the stationary phase and the liquid phase is the mobile phase. Substances are distributed between the stationary phase and the mobile phase. Cellulose layer is a filter paper, contains a moisture which acts as a stationary phase. Organic solvents or buffer are used as a mobile phase in the paper chromatography. Components of the sample will separate readily according to how strongly they absorb onto the stationary phase versus how readily they dissolve in the mobile phases. This is the paper chromatography diagram, and the next the procedures involved in the paper chromatography. First one, selecting a suitable type of development. It is based on the complexity of solvent, paper and mixture. Usually ascending type or radial paper chromatography is used as they are easy to perform. And the next, selecting a suitable filter paper. Filter paper is done based on the size of the pores and the sample quality. And the next third step is to prepare the sample. So, sample preparation includes the dissolution of the sample in a suitable solvent used in making the mobile phase. And then fourth step, spot the sample on the paper. Samples should be spotted at a proper position on the filter paper by using a capillary tube. And the next step, chromatogram development. Chromatogram development is spotted by immersing the paper in the mobile phase. Due to the capillary action of paper, the mobile phase moves over the sample on that paper. And the next step, paper drying and compound reduction. Once the chromatogram is developed, the paper is dried using an air dryer, and also detect the solution can be sprayed on the chromatogram developed paper and dried to identify the sample chromatogram spots. And then last, after drying the paper, we calculate the RF value. RF value means the distance travelled relative to the solvent is a constant for a particular compound as long as other parameters such as the type of paper or extent composition of the solvent are constant. The distance travelled relative to the solvent is known as RF value. RF means retardation factor. And the next, the types of paper chromatography. The various types of paper chromatography is available. First one, ascending paper chromatography. It is the solvent is moves in an upward direction. And the next, descending paper chromatography. The solvent moved to the capillary action due to the downwards. Ascending and descending paper chromatography. It is the both directions of particular point. And the next, radial or circular paper chromatography. The sample was deposited at the center of the circular filter paper and horizontally on a petri dish which contains the solvent. And the last, two-dimensional paper chromatography. Substance which have the same RF values can be resolved with the help of two-dimensional paper chromatography. And the last, applications of paper chromatography: to study the process of fermentation and ripening, and to check the purity of pharmaceutical, and to inspect cosmetics, to detect the adulterants and to detect the contaminants in drinks and foods, to examine the reaction mixtures in biochemical laboratories, to determine dopes or drugs in human and animals. So, these are the paper chromatography principle and the mechanisms. Thank you.